同学请迅速就坐哈。你看到有空的位置就坐。同学请迅速就坐哈。好，来吧，各位同学，今天那个庄所长有事不在，所以请我代理。那今天也是一个很好的日子，很好的时间，刚好今天的讲者是我邀请过来。好，这讲者叫做郭佩恩，他是目前这个他之前在 UC San Francisco，是医生，然后回到台湾就执掌中央研究院生医所的所长，相当不容易。反正你们看到我就知道哈，我邀请讲者都是很厉害、很厉害的那一种，所以呢，你们要懂得礼貌，好不好？那也希望各位呢，勇敢的发问问题。然后呢，上学期我们请交大校长过来，有问问题的同学都可以得到一餐，跟校长吃一餐饭，这是相当不错的。所以，我们可能下礼拜什么就会去交大吃饭。好，那个是好，那个所以呢，基本上今天呢是生医所跟电子所合办的 seminar， 所以你会看到这个有些朋友在此，因为上学期我举办了混合的，这是第2个学期举办混合的 audience， 因为上学期结果我觉得相当的好，所以这是我们在延续这个工作。好了，那个这个 title 什么东西都很多。好，那不如就请这个我们的所长中研院的所长来跟大家简介一下今天的题目，让我们以最热烈的掌声欢迎郭博士。
So thank you very much for having me here today. I was born in Hong Kong, grew up in Hong Kong, and I left for college when I was 18. So I can read Chinese, I can speak in Cantonese, but my Mandarin is really bad. So I'm going to speak in English today. But if you ask questions, you can ask me in Mandarin. I can understand 85%. Okay. So today I want to talk to you a little bit about a project that I would like to do in Taiwan. But it's based on DNA. And DNA, as you know, needs a lot of help. So today I want to tell you a little bit about what I do, and then I will tell you what I need you as young people who are really smart to do, okay? So about 7 years ago, the United States National Academy of Sciences started a meeting with experts. And the question that they asked was, how do we define diseases a little bit differently from what we are defining disease now? Because right now, we define disease by the organ and then the symptoms. For example, you can define something as lung cancer because it's in the lung. However, we realize that some cancers, even they are in different organs, they share very similar mechanisms. So this group decided that we should define diseases a little differently. Instead of defining them just by organ and by the symptoms, we should define them by the individual. Okay, so we are all different in this room, and we all have different characteristics. So if we can change the definition of disease from just the organ and the symptoms by the way we as an individual behave in the face of a disease, and then look at the disease mechanism, and we combine the two, then we can treat the diseases a little differently, especially if we know how we as individuals respond to different medicines. Okay, so in order to do that, this group says that we need to collect many sets of data, both individual data and also disease data. And then if we collect all these data, then when we look at a patient, we can look at the pattern based on all we know about other people and all the disease mechanisms. Then we can define this a lot more precisely, and we can treat it with individual treatment. So I want to illustrate this by a diagram. So if you think of health outcome, sometimes the health outcome is defined by the individual characteristics. So I'm a dermatologist. I look at skin and hair and nail, right? So this is a patient with no color because it does not have the enzyme tyrosinase that will change tyrosine to the pigment melanin. So this patient, whether you change his environment, whether you feed him different things, is not going to matter because he cannot make pigment. So no matter what you do, he's going to have white hair, he's going to have no eye color, and he's going to have trouble when the sun shines on his skin. He's going to have, DNA damage, no protection. However, there are other conditions. For example, this condition. So it's called guttate psoriasis. So this patient has DNA reasons why he will get this condition, but he's not going to get it unless his skin is challenged by an external factor. So this person gets the disease when he is exposed to strep. So he has a sore throat, and the strep bacteria, when there is enough of them, it will make a toxin that goes to the skin. Other people with the toxin, nothing happened. But this patient, because of his DNA, the toxin makes him blow up into this disease. So the way we treat this patient is to say, okay, this time it's too bad. But next time, when you have a sore throat, go get tested right away and get antibiotics, because if you don't have enough strep in your system, there's no toxin and there is no disease. So in this case, you have individual characteristics and external factors that cause the condition. And then finally, we have to think about the disease biology. Okay, so individual stuff, outside stuff, and the disease stuff. Okay, so the three things together define a disease. And so this is like when we use the map, the Google map, right? You can click on different layers of Google map. And actually people do this a lot, especially companies that try to target their customers. They think about the customer, where they live, what kind of environment they find themselves in, what kind of uses of the the land and whether they live in crowded areas or not so crowded areas, and they design a strategy to reach these customers. And so we can do the same thing with health. So if we find the people's DNA, the kind of microbiome they have, what kind of metabolites they have, what are they exposed to, what kind of behavior they have, whether they exercise a lot, whether they eat a lot of fat, or whether they eat a lot of sugar. And then we add all that together, and then we also get the history from the patient contributed data. Then we see the whole picture. So in the United States, we take a little bit more time to talk to the patient, but I hear that in Taiwan, the doctors don't have that much time. So it's going to be hard to get a lot of this stuff, but because of big data, we should be able to get a lot of that. So there are a lot of opportunities. So we have opportunities because a lot of the technologies now have become affordable. So we can collect a lot of biological data. And we also have electronic medical data. And then we can also collect data from devices. So how many of you wear watches and phones that collect data about your health and about how many steps you take and so forth? And then finally, people that you learn from, your professors and people who work in the computational sciences, they are using a lot of data mining methods that we can extract the data. But there are some challenges. There are challenges because the data that we want to collect have different properties. For example, your DNA is kind of static. It doesn't change too much. But there are data that change from time to time. For example, gene expression, or proteomics, or metabolomics, or microbiome. All these things change with the condition you're in, the stage of your development, and also whether you're well or you're sick. So these data, you cannot collect it just once. You have to collect it over time. And then we have to think about what kind of medical data we collect, and what kind of environmental data we collect, what kind of behavioral data you want to collect, and how to interpret the data and how to use the data. These are all very difficult problems. So just to illustrate the cost of getting DNA sequencing, when we did the Human Genome Project, it took $100 million to do one genome. But now you can get one genome done with 1,000 US. So it's a huge change in price. And because of this change in price, we can get a lot of nice data. So on the upper left is single cell gene expression RNA sequencing data. And if you sequence these cells from the blood, you can cluster them into different categories. The T cells, the B cells, all give you different patterns. And then, of course, we can get the bacterial data on our skin and on our gut. You can look at all the proteins and all the metabolites. And then, of course, you can get health data. So this is my data from I can go into my chart, called My Chart, in San Francisco, and find all my test results and the doctor's notes. And then there are these things that we wear. So there are devices on the right side. You can say, right now everybody knows where you are because your phone is being tracked. And my wife knows exactly where I am right now. And she's happy that I'm at Taida. So we can figure out the activity level. There are things that can monitor our oxygen level and what kind of heart rate we have, how much calories we have burned, and so forth. And people are doing additional sensors to look at blood pressure and air quality and blood sugar and so forth. So all these data can be collected in real time and sent to a database. So at UCSF, the cardiologists give out apps to all the patients with cell phones. And the app is tracking the patient. So if a patient is found at a hospital for more than 15 minutes, there's a text message to the patient saying, what are your symptoms? So they want to know whether someone is having a heart attack. So they actually send them messages like that. Okay, so, and then you guys, when you go to have dinner or lunch, you like something, you take a picture. And then you send it to Instagram or Facebook or whatever, and then all that is captured. And people actually know what you eat if you keep on doing this. And then all the different social media. And then there are people who send their DNA for testing. And all this become big data. So this is what you should think about. How do you find data from all these different sources of information? You probably hear about Netflix in the US, where we can stream movies. Every time you pick a movie, Netflix will tell you what other movies you should like. Same thing with Amazon. When I buy a book or buy an item, it will tell me what to do next. And then there's Target. So Target is a department store in the US. So I told this joke before, but a few years ago, the people who work for Target came up with an algorithm to find out whether someone is pregnant or not. So now, for the young people in the room, what do you think the identifying feature of someone who is pregnant before they even show? Do you know? Well, Target knows, because Target has been tracking how people spend their money. And they've realized that when a woman is pregnant, they stop buying really fragrant body lotions. They totally stop buying those. They buy no fragrant lotions. And so as soon as they figure that out, they start sending coupons to the women who switch from really good smelling lotion to plain lotion. And then they can actually predict when the baby is born just by the switch. So now if they can do that, we should be able to figure out health by looking at big data. So that's your challenge, okay? So this is the group that will solve that problem for us. And I'm hoping that you will do it. Okay, so I'll tell you a little bit about a couple of projects that we do in our lab. So I wanted to look at germline properties because it's static. I only have to do it once for someone's life. And it's still very expensive to do if we do it for everyone. And so the reason why it's expensive to do when we look at complete analysis is because our DNA is like a big puzzle. So this is the world's largest puzzle. It has about 33,000 pieces. And some guy in the United Kingdom solved it with a bunch of his friends over 2 weeks. And then now it's hanging at some children's hospital near his house. You know why this is difficult? It's difficult because there are so many pieces. And so many pieces are exactly the same. All the leaves, they're exactly the same. It's like a repeat. So you cannot solve it without knowing the shape. So in a puzzle, it's good because every piece is a unique shape. But in DNA, it's not so easy because in DNA, we have 3 billion bases and there are many, many repeats. And so in fact, half of our DNA is made up of repeats. That means that there are very, very similar sequences in multiple places in the genome. And so right now, when we do our sequencing so cheap, is because we break the DNA into little pieces and we sequence 150 bases at a time. Well, 150 bases, you have so many pieces across the genome, and because of the repeats, you can only line them up uniquely for about 30,000 bases at a time. Because every 30,000 bases, you hit a place that the 150 base read length will not cross over. So because of that, now you have 100,000 pieces to try to put together. And that's not easy. So I don't know whether you have thought about this problem. We need help. And so nowadays people say, oh, we have sequenced a lot of people. Well, they have, but they have not completely sequenced them, because all they have done is to align the reads back to the reference. You get that? So you're aligning to the reference. You don't have a complete genome every time you sequence. So the $1,000 genome gives you a bunch of reads that you can align back to the reference. Well, the problem is that the reference is not perfect. In fact, the reference is not even made of people from Asia. The reference is made from people from Europe and a few African Americans. That's it. And actually, we have done a lot of sequencing of Asians, and there are many sequences that we find in Asians that are not part of the reference. So when you try to align your sequence back to the reference, you're aligning it to an imperfect model. So what is missing? We're missing structural variations, and we're missing phasing information, meaning variants that are near each other. You don't know whether they're on the same chromosome or they're on different chromosome. So if you look at this chart, you'll see that there are different kinds of structural variations. So if you have the reference on top and then you have someone on the bottom, sometimes some of the DNA in the reference is missing in the individual. But then there are other times, like when you have the reference, but then you're sequencing someone who is not a European or not African, you see pieces of DNA that are in there but not in the reference. And then there are all these mobile elements. The repeats that come from the mobile elements that our DNA incorporated. They're mostly of viral origin or other, we call transposons and so forth. And they will insert all over the place. Sometimes, because of these mobile elements, the DNA will expand, and sometimes they get duplicated, and sometimes they get inverted, and sometimes they get translocated. Because the way to get variation in our genome, there are two different ways. So one is to have a mutation. So it's a one base at a time. When we have germline mutations, then you see different bases. But there are also variation because of recombination. So when our sperms and our eggs are formed, the two chromosomes from our parents come together and they recombine. And so this is all based on recombination. And because of this, actually there are a lot of our DNA change because of this. So I don't know whether you have taken biology classes where your teacher tells you that our DNA is 99.9% identical. So that's only based on the single base changes. But if you think of the changes due to structural movement of pieces of DNA along the chromosome, is actually different. So about 4% of the human genome actually is based on this variation. And so the bottom line is that we need to do something to access this kind of variation that is not available when you only use whole genome sequencing. And so phasing. Phasing is talking about variants that are along the DNA. What is their relationship when you have multiple variants? Well, if the top is the chromosome from your mom and the bottom is the one from your dad, and you have all these different variants. So when they're together, sometimes they can act in a way that change the gene function. But when we sequence, we break them into little pieces. So after you have done the sequencing, how do you put it back together? When you put it back together, it could be many different ways. So when you have different combination, it may not reflect what the original relationship is. And so phasing is a big problem for short sequencing reads. And so in order to take care of this, we need a much better sequencing method. So right now the sequencing method only gives you 150 bases. Well, if you have a much better sequencing method, you can sequence the whole chromosome from end to end. This is what our body does. Every time we have a duplication of our cells, our body can do the DNA from end to end accurately. So all your cells, like every 3 weeks, your skin turn over, your blood turn over, and they can copy this easily, no problem. But we don't have a way to do that yet. And you want to be really accurate, and you want to be really cheap, really easy to do. So far, there's nothing we can do that. So, how can we get the whole genome assembled back without this ideal sequencing method? And so what you have to do is to make the puzzle small, with bigger pieces, smaller pieces of, smaller number of puzzle pieces. And so this is what our lab has done over the last few years. We figure out a way to do genome mapping using really long DNA, which is replacing the bacterial artificial chromosomes that the genome project used in the beginning. And then we adopted a new long sequencing method to replace the shorter clones that the genome project used. And so these are the two technologies. They are both commercially available. The first one is by a company called BioNano Genomics. It's a Sapphire system that can put long DNA pieces through nanochannels and take a picture and map it. And then the next is a company called 10x Genomics that can put barcodes onto DNA pieces that are pretty long, and then when you break it apart, the barcode tells you that the different reads come together from the same molecule. So this is the method we published a few years ago. So what we did was, instead of breaking the DNA using a restriction enzyme that recognizes a motif, we use an enzyme that is mutated so that it will only break the first strand but not the second strand. So when you have that, then you keep the DNA intact, and all you have to do is to fill it in with a fluorescent dye. So once we have that, if we can measure the distance between the different fluorescent tags, then we have a map. So I'm going to show you a movie. So this is the DNA going through the nanochannels. So you can see it goes through, and then there are all these little packs that allows it to go around. Now this is individual DNA molecules. So then it goes all the way around to the bottom, where it's put into this really narrow nanochannel, so that the DNA is stretched in a uniform way. So once we have that, then we can put it on a, so this is what the chip looks like, and that's the machine. And so once you have that, then you can actually look at the image. So you have all these molecules, each one about 200 to 500,000 bases long, and then you can put the fluorescent dye tagging onto it. And then once you have that, it's actually a very easy algorithm, because all you have to do is just measure the distance. So every molecule is translated to a string of numbers. So how long and where the markings are. So once you have that, then you can line them up and give you these contigs. And because the molecules are so big, that you can actually find things that are not seen in the reference. So for example, the green bar is the reference. And you can see the pink area is where the reference genome has 6 copies of this repeat. But then in our sample, the blue one, there are actually like 14 copies. And why we know there's 14 copies? Because we have big molecules that go all the way across. So there is no way that we're going to make a mistake, because we see big molecules. And here's another example of a big insertion. The reference doesn't even have this. But because we have big and long molecules, we know exactly where it is and how much is inserted. And then if it's an inversion, it's actually very easy to see, because we can tell the constant part, the unique part on one side, unique mark on the other side, but the pattern is different from what the reference says it should be. And so with this method, we can bridge across all the repeats that are smaller than our molecule size, and we can come up with a really nice map. So now the other technology by 10x Genomics is very interesting. So the color circles are different barcodes on a bead. And then every bead that comes through this microfluidic device, you add enzyme, you add long DNA, you add some oil. So then the oil traps it into a little, little droplet. And then in that droplet, because you have the enzyme, you have the DNA, you have the barcode, then you can make, amplify it into little pieces, each with a barcode. And then after you've done this reaction, then you can break the oil and pull them all together and make a library so that you can sequence it. So after the sequencing, even though the reads are still 150 bases, but because each of them with the same barcode, came from the same molecule, then you can put them all together, and now you have a nice long contig that even if you have repeats, you can go across it. So now, because of DNA sequencing is using the Illumina machine, so it's very accurate. And so now you can actually find the breakpoints of all the different structural variations. Okay, so we have done exactly that. So we have published this a couple of years ago. One of the samples that people have used a lot. So if you look at the top is just, if you just use the DNA sequencing data from the short read machines, that's all you get, little pieces. But then the middle is the 10x genomics assembly. You can see that some of them are actually very big. And then if you add to that the bio-nano maps, then the two together give you a really nice hybrid map that covers a really long distance. And so we came up with a map of 170 pieces. So as you know, we have 23 pairs of chromosomes. So a perfect map has 46 maps. Okay, one chromosome arm, 2 arms per each chromosome, 23 pairs. So if it's a perfect map, there should be 46. So we're not quite perfect, but we're pretty good. And so with this, so each color bar is one contig. So you can have really nice contig size, and then when you go down and use all the single base changes across the genome, you can actually phase them. So now in one experiment, you can get all that information. So this is really nice. So we can combine the different technologies, and we can do everything. And right now, it's about $5,000 US. Okay, so it's 5 times more than you just do the regular shotgun sequencing, but you get all this information that you'll never get for just the regular sequencing experiment. Now, this is still quite expensive, so we're not going to do that for everyone. But we're going to do this as reference. So what we're doing now is to sequence a bunch of people in Taiwan, and we're going to make this our Taiwan reference genome. So you don't have to go back and use the Genome Project genome, because it's not for Asians. And this is going to be really good for us, because now you have something that you can compare to, and you know exactly where the structural variations are, and you know exactly where things are. And the reason why we can do that is because of this thing about population genetics, because Taiwan came from a small group of founders. And these founders leave a legacy in our DNA. And if we sequence enough people, we will capture all of that. And then once we capture all of that, you never have to sequence again. Unless you have cancer. Or unless you have a really bad genetic disease. But for the rest of us, we don't need to sequence. Okay, so there are some challenges, because we want to keep the DNA long, so we need to use fresh cells. We cannot use the DNA you store in the lab that has been extracted by other means. And also, even with the 10x linked reads, if there are repeats within the molecule that are longer than 150 bases, then the placement is still not unique. Okay, so then once we have done that, then we want to see, well, if we can map all the structural variations, what does it look like across the world? And so we picked out 26 populations that the 1,000 Genomes Project used, and we picked 6 samples from each of these populations. And we mapped them all, so 150 some samples, and these are the observations. So we observed that there are a whole bunch of sequences that are in our samples across the world that are not in the reference, which is not a surprise, but we know that. And then we also find that a bunch of sequences from the reference not found anywhere else in the world. So these are unique reference sequences that are not found in the rest of the world. And then we also found about 100 really complex regions that is different in terms of the way it evolves than the regular genome. And I'll tell you a little bit about that later. Okay, so what does that mean? So what it means is that if you look at the chromosomes on the left, there are different kinds of regions in our chromosome. So there is a region like B, where it's very, very simple. If you look at it, everybody looks alike. So every line is a different person. They all pretty much look the same. And then there is something like C, where, yeah, they're not the same from person to person, but there are only a few patterns. But then there is the complex regions, where everyone is a little different. And the reason why this is so is because this region is full of repeats, so that every time you have a baby, the recombination happens, very high frequency. So it's revolving at a much faster rate than the rest of the genome. So if we look at if we ignore the complex regions, and we just look at the regular regions of the genome, you can cluster the different populations this way. So the red, the Africans, the green, East Asians, including Taiwan and China and Japan, and then the Europeans and the South Asians and the Americans up on the other side. And this looks exactly like what you do if you have the SNPs, the single base changes. So telling us that the regular part of the genome and the single base changes have very similar rate of change, rate of evolution. All right, so the complex regions, we're in trouble because they are full of so many repeats that we can't really overcome them. And so they are near the telomere, which is the end of the chromosome, or they're near the centromere in the middle, or in regions called segmental duplications. And after we have looked at 150 people, we realized that the reference genome is just one of many, many haplotypes. So if you use that as your reference, you can't make sense of it. So here's an example. So this is an area called the pepsinogen A cluster. And you can see that on the left and on the right, there are really clean, very common patterns. But in the middle, in the black part, you see that some people, each line is one person. Some people have more copies of these colorful motifs than others. And if you count them all up, it turns out that Asians have more of the copies of this repeat than others. And I don't know how many of you know that this region actually is implicated in gastric cancer, stomach cancer. And we all know that Asians have a higher incidence of stomach cancer. So whether this is true or not, or related or not, I don't know. But there are patterns of different populations. And then here's an even more difficult region, chromosome 21. So B tells you what the reference says. But in fact, after we have looked at many, many people, it's more like this. And so this tells us that for some parts of the genome, we just cannot rely on the reference. We need to look at a whole bunch of people together to figure out what's what. Okay, so that's the end of the technical stuff about DNA. But you need to know, because if you don't know this, your algorithms will be wrong. And so, if we know all this, then what is the opportunity for Taiwan? Well, Taiwan has some really good stuff going on for it. One is that it's a pretty homogeneous population, because if you look around the room, most people look alike. And we have very similar population history. So because of that, we can actually construct a bunch of reference genomes. And it will capture most of the diversity in the country. And then, because of that, we can make a Taiwan-specific SNP array. Now, the other thing is that Taiwan has national health care, so that it's one payer that can allocate resources. If we can add genetic information to the medical record, we can actually do something really useful. And of course, we have really good doctors here, and really good hospitals, and nice medical records. And because of the nice records, we can actually look at the patient's complete medical history, and also we can have long-term follow-up. So what we're doing now is that we are starting a project called the Taiwan Precision Medicine Initiative. And so this is already, we got funding to do this. So first we're going to make high quality references. So it's done at the Academia Sinica in my lab. So we're gonna do this by this summer, because of this easy way that we can make maps and do sequencing. And then we're designing a custom SNP array for Taiwan. So this one has all the mapping variants, plus all the known genetic variations that cause disease in Taiwan that we can design. And then we're gonna genotype 1 million people across the country in the next 3 years. And we genotype them not just for the sake of genotyping, but we want to use the information to help in their patient care. And then we're building a research database that can be used for genetic analysis. And we're doing this because sequencing is still expensive, so we can't afford sequencing 23 million people. But I want to say that we can genotype 23 million people, because right now we negotiated a deal that we can do a genotyping test for 1,000 NT. So now 1,000 NT is like less than a lot of blood tests that we do in the clinic. So if we can use this price, then all you have to do is to do one less other blood test in your lifetime. You can pay for it. So I get a physical every couple of years, and my doctor always takes some blood and do my cholesterol level. I just do one fewer cholesterol test. We can pay for this test, and you have this data forever. So that's what I'm trying to convince the hospitals and the government to pay for this. And so I know that we're running out of time, so I will go really fast. And so the reason why we can use single base changes on an array is because if we have the reference and we know the population structure, we have all the haplotypes, then we can impute based on the reference. If you know, say, 300,000 variants in your genome, we can impute the rest of the sequence from that if we have the right set of reference genomes. Now, all the rare, all the new mutations we cannot find. But for all the common stuff, we can get it. is actually very accurate and very fast. So for common diseases, it's not some rare thing that happened in our gene that make us so, but it's because of a combination of variants that are common in the population that make us susceptible for certain disease. And so that's what we're going to do. So for cancer and for really rare diseases, we have to sequence. But for 90% of the population, this is enough. Okay? All right. So this week, we're going to be done with the design. So we're going to have a SNP array with about 750,000 markers that collects all this information. So once we have this, we can find out what your blood type is. We can do your HLA typing. We can find out whether you're a carrier for some of the common variants that cause disease in Taiwan. And also, we can figure out whether you are allergic to a drug or whether you are going to have a bad response to a drug. Okay, it's not perfect, but we're going to get a lot of information. So we know that it's going to work because of the project I did 7 years ago in San Francisco. So there is a healthcare system in the United States called Kaiser Permanente. So Kaiser is like the national health system here. They keep all the records, they do all the tests, they give all the medicines, and so forth. And so we genotyped 100,000 people, and we did it in 14 months. And we generated 70 billion genotypes. And so with that information, just show you something really simple. So this is a graph that tells you how our drug acts in our body. So most of the time we want our drug to be within the yellow zone, which is a therapeutic zone. So that's why you have to take drugs sometimes twice a day, sometimes 3 times a day, sometimes 4 times a day, is to maintain your drug level at the right place. But if you have a gene that cannot metabolize the drug, and you're getting the same dose as your friend, then sometimes you will get into the toxic dose because your body is not eliminating it fast enough. And sometimes it's all the time you're in trouble. So when we did the 100,000 people, we just asked. So the US FDA asked us to do a genetic test for 150 medicines when we give patients. So since we have the data, we just say, okay, if we just look at these genes, what happened? Well, what happens is that 90% of us, or 90% of the people that we genotype, have at least one drug that they're in trouble, at least one gene that they're in trouble. So this is actually very telling. So you see the column that says ASN, which is Asian. So all the drugs were developed in the US or Europe. So Chinese are in trouble because all these drugs, we're always in trouble because they never tested us when they developed the drug. And so based on this data, 2/3 of the people with these genes that cannot metabolize the drugs properly, they were actually given the prescription for these drugs. And 17% of them were actually having the high-risk genotype, and they also got that drug. So now 17% of 100,000 people is 17,000 people. So if you think about 23 million people in Taiwan, it's a lot. So if we don't do anything else, this chip will help us take care of this problem. Okay? So this is what's going to happen. So we're working with, hopefully, some of you to come up with a way that when the genotype is stored in the medical record, when a doctor clicks on the order button to order a medicine, something will come out and say, oh, for this patient, you have to change the dose. Or for this patient, you have to try a different drug. And so this is actually not new. Y.T. Chen at Academia Sinica already identified several HLI types that cause really bad reactions when people take seizure medications or pain medications. And this is paid for by the health system to test people before they take these drugs. But with our chip, this is done for everyone before they ever come to the clinic. Okay, so then we want to do this as a clinical test rather than a research test, because when we do that, we can actually use the actionable content on the array in clinical practice. And how do we use this? Besides preventing drug reaction, we can actually use that to tailor management. So what am I saying? What I'm saying is that because of that same study, we actually asked a really simple question. We say, if we know all the risk variants for cholesterol level, we just add them up, 1 point for every variant that the patient has. If we add that point, all those points together, and give a score, how does that correlate with the time the patient is prescribed an anti-cholesterol drug? So I didn't even look at the cholesterol level. All we say is, what is the age that the person is given statin? And amazingly enough, it's highly correlated. So that means that if someone has a high score, then they will have high cholesterol very young. When someone has a low score, much later. So if that's true, then we can change the way we manage our patients. Because right now, the recommendation is that anyone 35 or older gets a cholesterol test. Well, but if we know the score ahead of time, then we just say, well, if you have a low score, then wait till you're 50. When you have a high score, start young. So this way, we can really prevent heart disease of the people who are susceptible. And we save a lot of money because most people don't have that problem until later on. So we can do the same thing with cancer risk, we can do the same thing with stroke risk, and we can do the same thing for metabolic risk. Now this is not perfect because our prediction is not perfect. But if we have millions of people with this information, then we can make this more perfect. So that's why we need to start big, not small. Now we still need to sequence. We need to sequence cancer, we need to sequence rare diseases. And so we're doing this. So NTU Hospital has an undiagnosed disease project. So they do whole exome sequencing to find causal mutations. When they don't find the diagnosis, we are collaborating with them. We take all their unsolved cases, and we're going to do our full genome analysis with it, so that we can find changes that you cannot see by exome sequencing. So here's a case that we did in the U.S. So there's a 2-year-old boy from Yemen. The parents are related, so that they have a lot of homozygous regions, and the exome is negative. And he has neuroblastoma, which is a brain cancer. So we did the test. So this is just based on the barcode sharing of the reads. When we look at that, the parents are, when we look at chromosome 1 and chromosome 9, nothing. But the kid has an area where the barcode goes from chromosome 9 to chromosome 1. So it turns out that we can find the breakpoint of the translocation, and it affects a gene that causes neuroblastoma. And so this is the map. You can tell that the green is the reference, the blue is the test case, and you can see that for one of the chromosomes, there's a change that goes from there to here. And so we just ask, okay, well, let's do a karyotype. So when we look at the chromosomes, we can see the change, moving one part of 1 to the other part of 9. And so this is a really easy case for us to solve, and it helps the patient figure out what's going on. Okay, so this is probably one of the last slides. So this is what I want you to take home. Since you're not biologists, you need to know this. So one is that precision medicine means that everyone has to do it. So if you don't do it, we have nothing on you. So you cannot use someone else's data for yourself. You have to have your own, okay? And number 2, we need to have many, many sets of data in order to really make this powerful. And this is all based on cost. So if the cost is cheap, everybody has a cell phone now. 10 years ago, nobody has a cell phone. But now you know exactly how many steps you take every day before you know nothing. And then the other thing has to do with interpreting the data. How do we interpret medical data? How do we interpret biological data and so forth? And then the final thing that we want to be able to do is to collect the right data at the right time and monitor the data continuously so that we can tailor the treatment and adjust the treatment in real time. So I thank you for your attention. I'm happy to answer any questions.
同学，我先问你们一个问题好了，你们有正在做 DNA 的研究，可以举个手吗？可以站起来一下吗？主任，来站起来。对不起，有，一定有啦，你们，同学们站起来。那个好，应该不止，你先做，举个手好不好？再举一下手。好，那个有兴趣到中研院的为所长弄出，待会可以跟所长联络一下，好不好？那现在就开放大家再问问题啊，这个难得的机会哦。好，请。
Yeah, you can ask in Mandarin, okay? No one is completely healthy. That's easy. Perfect. Well, so that's a very, very good question. But the one thing about genetics you need to know has to do with our environment. So you have a perfect gene for this environment, but if you go to Alaska, you go to Saudi Arabia, you maybe have a very, very bad gene. You know what I'm saying? So it really depends on where you find yourself. So there's I think the more senior members of the audience will remember, for the longest time we think the Japanese don't have heart disease. It's because they live in Japan, they eat rice, and they have very calm lifestyle. Guess what? All the Japanese that move to America, they all have heart disease at the same rate as the rest because they're eating Big Macs, they're having really stressful lifestyle, and they have exactly the same prevalence of heart disease. So your question is, depends on where you live and depends on what you do. And so there may be perfect genes for people who are very sedentary, and then there are perfect genes for people who are really active. So I'm hoping that, I don't know whether you know a really old comedian or a movie star who passed away not too long ago. He lived to 100. His name is George Burns. He lived to 100. He smokes, he drinks, he fools around with women. He does everything. And everybody wants his genes because he can do anything bad and still healthy.
在座有2位超级的重要人物，我们请。
那个我有另外一个会，所以迟到了。不过我想前面部分比较偏重这个技术的部分哈。那我本身是台大院医师，我们院长坐在那里。我想那个郭所长他的最大的 impact 是他有这些技术，刚才他讲得很清楚，可以用比较低的价钱呢，可以告诉我们一个人类的，当然也可以用到动物了哈。不过我们是医院，主要是不医院也有兽医院哈，我们是台大医院，所以是人。那过去我们做很多 test， 都是想要了解这个病人他什么病。它怎么样发生的？那我要用什么药？可是这些有一些限制，到某一个程度呢，你一定要看它的基因的这个变化。那郭所长现在的这个带进来的这个技术呢，还有他的知识呢， 就可以解决这个问题。所以我个人是觉得，尤其台湾这个 population size， 还有我们有很好的这个 system 嘛，我个人是认为是这个将来是非常 promising 的一个 field 哦。那也就是因为这样，我们才千辛万苦把郭所长请回来台湾，这个非常不容易的事情啊。我正好是中研院的那个他的所长的 Search Committee 的 Chair， 所以我想非常了解他的这个工作啊。那所以呢，我想这些工作需要除了医药那一端以外呢，需要前面的很多的帮忙。那最多的可能的人才呢，就是可能出自于我们的电资及资讯学院的这些年轻人。那也许你们还太年轻，也许老师知道这个事情。电资学院以前是不做跟生物有关系的，直到差不多10年前吧，郭伯文当所长，院长的时候，许伯文说错了，不是，他姓郭，这个乱 recombination 乱接一通，那我那个时候因为许院长呢坐在我行政会坐在我是医学院院长那个时候，坐在我的右边，我就说，他跟我在闲谈其他事情，我说你们电子学院不错哦，很厉害哦，但是我说你们有个缺点，他一听啊，我电子学院那么好，还有缺点呢，他说是什么？我说你没有生物有关系的，医学有关系的，他听进去了，所以你们学院才开始有做生医的老师啊。那我想这个方向是对的，因为后来我们看到贵院有很多的进步。可是呢，我想现在进到基因的一个层次呢，我想我们需要新血来加进去。那个郭所长今天来介绍这个是非常非常不错哈。我大概只能 comment 到这里。
可以，可以听，不可以讲。
我们再请院长，谢谢他们。
很多人想要做 precision medicine， 所以他有一个 reference。但是过去大概郭教授刚刚也讲过，很多人都用 sequence， 但是常常我们医师拿到那个 data， 病人问我们说这代表什么意思？我们说我也不晓得。所以这是一个大问题了。那今天我听到郭教授的方法，大概不是 perfect， 但是可以解决一部分的问题。那其实台湾在整个这个genome的 study 也是这个 human genome 开始以后，其实影响很多了。第一个大概影响的就是这 sequence 的 technique。还有它的 cost， 很明显的 technique 变快、变好、变准确，然后它的 cost 一直 down， 那个 machine 也越来越小，所以是很好的。那再加上，其实这个要很谢谢电子学院的这个 big data 的 processing 跟它的 mining 或者是什么，这个完全是各位的帮忙才会让这个human genome 进步这么快但是还是有很多问题那现在应用最多了，大概是在 medicine 上最多的，大家想要说会不会有 individualized medicine， 就是个人化的医疗因为我们在刚刚郭教授也提过很多的治疗大概那个药物的反应，特别是 target therapy 或者是 cancer 的病人，变成你用的药物要越来越精准。这个 population 里面也许10个有效，但是90个没有效。但是换到另一个 population， 90个有效，10个没效。所以这个 这个药就变成非常的需要这些治疗，而且你有效，你哥哥不一定有效。我举一个例子哈，我们曾经把一个 family 的那个先天性心脏病做成 iPS， 然后我们去看，所有的基因都一样啊 ，mutation 一样啊，可是它在 even 在 disk， 它的 response 不一样，所以这个就变成后来有 epigenetic， 有各式各样啊。最近的 paper 甚至于认为说 protein 可以去改变那个基因的 3D structure， 就可以改变它的 expression 跟它的方形，类似这样。但是这个都要各位的帮忙。这有点像刚刚郭教授也提到，现在 Google 看一下，做一下分析的话，就可以说啊，1个礼拜以后大概流行感冒会很严重，所以医院要开始注意。我们现在我们的卫福部就是利用急诊或者是大家上 Google 查感冒药或发烧怎么处理，就可以知道接下来会发生什么事，类似这样的哈。不过我要问郭教授的几个问题，就是说，其实中研院已经开始所谓的 biobank 跟 biosignature， 但是用的方法不一样嘛。你现在的方法也许会好，但是那个 project 现在怎么跟这个连在一起？还有过去收集的那些资料，或者是那些 sample， 你刚刚有提过嘛？那是经过 processing， 所以你要的 sample 是 fresh， 可能不太一样。所以是不是代表说你这个 project 一开始那边的就要变成另外一种方式？你的会有没有什么不一样的？
No, actually, we use the biobank data for the reference. So we actually, the biobank sequenced a whole bunch of people. So based on the sequencing data, we know the principal component analysis. So we pick the right people for the reference.
Okay.
So that's number one. And they kept cells, so we actually can use their cells to do this. And we are working with them to come up with the chip. And so the design is a little different. So the biobank design is a very kind of uniform assessment of the patients. And then the genetic data is only one piece. Whereas we're using a cohort design, which is a totally different idea. So the two are very complementary. So we definitely, actually, we work together really carefully.
Okay.
And we will make them better, and they'll make us better.
Yeah, that sounds good. 2 more questions. Don't forget the mitochondrial DNA.
Yes, it's on the chip.
So it's also on the chip. 我刚问的就是说，人类的基农除了刚刚讲的核的基农以外，在我们的粒线体也带有30几个基因，这30几个基因其实在现在的 disease 或者什么，它是重要的。因为你可能跟你的哥哥一样，或者是你的 identical twin 是一样的，但是在某些变化的时候，又环境的影响，mitochondrial DNA 会变化，我也不晓得。听说那个太空人 twin，identical twin， 到外太空去回来以后，有 7% 基因会变。它到底是怎么变化，不晓得哈。那另外也要一个问题，就是说，台湾虽然是一个比较 homogeneous 的 population， 但是我们有 Aboriginal， 那是 South Island 的基农，那也有从中原时代的北方来的，那也有从 migration 的时候从东南亚来的。那过去大家认为说人类的演化是只有一个 origin 从 Africa， 但是现在发现其实是不同的，在基农的 inbred 或 outbred 或 mix 那这个会不会影响你的 project？ 因为原先的 biobank 是有分，所以它有一部分是来自我们的原住民，有一部分是来自哈卡，有一部分是来自汉人，所以你的这个也是会包括这一些。
Yeah, we are capturing all of that in our reference set. We just don't know whether. So we're betting that 500 is enough. May not be enough, but we'll start with 500.
OK， 因为这个其实是很重要的哈。同样一个病，在这3个不同的人要算种族还是什么 population， 它的表现是不太一样。
Yeah.
而且它的 even 光喝酒这一件事，对这3种人的健康的，或者是我们陈院长最清楚的那个 liver disease 都有不同的变化。
Right.
谢谢你的。
Those are very important questions, and we're trying to address all of them.
我想要那个请你吃一餐饭啊。我们来猜一下哈，猜这个同学可以到中研院跟那个院长吃一餐饭哈。来，你们想好哈，这个他的年龄是4字头的，举手。你们看一下，4字头举手，5字头的举手，6字头的举手。那位有。好，待会来跟我登记一下，这是很可怕的事情。这个同学们，你们可以开始做基因研究。如果能够基因再配合皮肤科，能够达到这样的东西，我想呢，这是一个真正的奇迹啊！来吧，那今天就时间快到了嘛。好了，那个来同学就我们谢谢今天演讲者哈，对。陈院长跟那个何院长，请留步。请留步。对不起，有点泄露你的年龄。